Sure.
Okay, Father, Son, Holy Spirit. Amen. God, grant me the serenity to accept the things I cannot change, courage to change the things I can, wisdom to know the difference. Father, Son, Holy Spirit. Amen. So I had a request, I had a couple emails asking me if we could see your quizzes. What I will do is we'll go over the questions here this morning real quickly. And then if you want to look at them, we can look at them on a Zoom meeting. All right. It's better to do that in case you have other questions. Okay. All right. There we go. Awesome. Okay. Commonly missed. This one was probably one of the most commonly missed questions. You don't look at the shape. What do? You count the carbons. Ready? I can't do it. I'll use my folder. And all my papers will fall out. Ready? 6 hexos. The shape doesn't tell you everything. Hexos. Actually, since this is a hexos, I know this is fructose. Yo-os. Okay. Secondary structure of protein results from the formation of hydrogen bonds. We hit that and we hit that. Awesome. That's great. Struction. That's when structure and function work together. We call that struction. I just made a new word. The struction of a triglyceride is energy storage, whereas the principal function of a phospholipid is plasma membrane, C. Which of the following is not part of a cytoskeleton? Okay. One of these is part of a plant. Centriole, found in plants. Everything else, those are small little proteins forming the cytoskeleton, which is, what kind of cells actually have cytoskeleton? Prokaryotic or eukaryotic? Marie. You. That's right, because it's all about me, not you. That's right. Good. So centriole, that also was a commonly missed question. So two of the most commonly missed questions. Number 1, number 4. From your reading, number 4 was a little bit. I think I even warned you ahead of time saying you better know these. Didn't I? I did. Oh, he's not really going to ask that, is he? He told me he would. All right. I'll stop now. Eukaryotes, prokaryotes, both. Pro, right? Because if it's in a eukaryote, it needs to have an us, right? You and us are related. Ribosomes, both kind of cells need to make proteins. Cell wall, both. Endomembrane system, eukaryotic. Good. All right, this one. If you put the specific molecule, which is glycerol, you got it right. If you also said hydrocarbon, I said okay. All right, if you said hydrocarbon, you also got credit for that. Sweet. Next. All right, we've been doing this drill for 5 weeks now. Okay, 3 weeks now, we've been doing this. Every time we see something, when you see a molecule, don't just say, oh, that's a new molecule. What kind of molecule is it? What kind of monomers make it? What kind of bonds holding it together? What kind of function might it have in my body? When you're eating that bagel eaters. All right, we're going to talk about bagels a little bit later today. Don't just be like bagel and body. Be a geek. Go all in. It's a polypeptide. What's that tell me? Polymer is protein. Bam! Everything else follows from there. Monomers are held together with a peptide. Oh, is it a monosaccharide? No. What's the monomer? Amino acid. Monomers for ADH are held together by a peptide bond. Absolutely. Hey, yo, Adrian. The initial synthesis begins in the cytosol. Initial synthesis, translation of proteins, always begins in the cytosol. I think I said that like 300 times on Friday. It's completed in the smooth endoplasmic reticulum. No, where's it completed, Cam? It is, but which endoplasmic reticulum? Perhaps in the rough if it has to go somewhere. Actually, this one would, because I tell you, it's released by cells in the pituitary gland. Do you guys notice that? I gave you clues. So part of some of my questions, I'm going to give you clues about this molecule. So sometimes when we miss these questions, it's because we miss details. It's released. After processing, the ADH is packaged before blah, 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 plasma membrane that synthesizes and releases it. Yes, right? That's true. Golgi. Post office of the brain, or Google the brain, whatever you guys want to call that now. Server of the brain. Okay. Everybody got 1, 2, and 4 pretty much. Most people got 1. Those are beta glucose molecules. That's carbohydrate. What's number 2? Nucleic acid, specifically. But we're going to talk about that, I think, today. This is the one that caused everybody problems. One of the most common answers I got for this was protein, because you're seeing these ring structures here. Oh, that looks like maybe tryptophan or something. What is this? 4-ring backbone. Jared? This is a lipid, specifically steroid. When you see the rings, be the Lord of the Rings. You want to get strong and buff, man. You know what I mean? Yo. This one. What about here? What about here? What's this one? That's also a lipid, specifically a triglyceride, which has a function of energy storage. See how fun this is? And these are all what kind of saturated fatty acids, which means that at a room temperature, this is going to form what we call beautiful. Yeah. This makes me happy. I'm going to sleep all tonight. You guys are making me happy. Okay, this question was kind of a train wreck. Not going to lie. So, I definitely forewarned everybody about this one, but that's okay. So let's go over these. B is the polar heads. A, the non-polar tails, right? Now, one of the things that may have confused everybody is that these amino acids, the R group is on the bottom instead of on the top. So I was being very tricky. No, I wasn't. I'm just putting a molecule up here. It's part of the deal. R group is down here. I could have had them flipped and flopped all over the place. So, what kind of an R group is this? Non-polar. Non-polar? Good. It's all hydrocarbons. Where am I going to find it, John? Boom. Boom. Good. How about this one? How about this one? Sam. Non-polar, right? Hydrocarbon, 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 hydrocarbon. Hey, let's do the hydrocarbon. Yo, what's that look like? I don't know. All right, where's that go? Kyle. In the middle. Yeah, hydrocarbons. Hydrocarbons, they stick together, right? They're like friends forever. What's that interaction called whenever those hydrocarbons are coming together? Back here in the corner. Hydrocarbons all coming together, pushing the water out. We call that what? Hydrophobic exclusion. Get the water out. Yo, sweet. Phenylalanine. No, what is that? I don't know. I think it is phenylalanine. What about this one? Kristen. Why? Right here. What kind of functional group is that? Antonia? Okay, good. So we got this polar group here at the end. So this is going to be where might we find this one then? Callum. Okay. Where might we find this amino acid in a protein that's in the plasma membrane? B or A? Polar. Good. All right. So if it's polar, it's going to be in B. What about 4? A or B? B. Okay. Good. Next page. Yeah. That one. Got to know how they're related. Through dehydration synthesis, a series of dehydration synthesis reactions. Glucose monomers are bound together to form the polymer glycogen to store energy, something like that. Or you could start with glycogen, break it down. We're going to do that today. All right, questions? Okay, we got to move. It's 1:45. So if you want to look at your quiz, at specific questions, send me an email. We'll do something online. I'll share the screen. We can go over your questions. Good. That's what we're going to do. If you want to look at them, you contact me. Okay? That's how it works. Okay. TV land, what are you guys seeing right now?
I'm seeing SimPorter.
Sweet. Thank you. Thank you, Matthew. All right. So quick review. So we talked about active transport, right? Anytime a molecule is moved from a region of low concentration to high concentration, that's always going to be active transport. At some point in the process, ATP was used. At some point. So requires the use of energy. We ended up sort of rushing through, so I want to go over this real quickly. We rushed through it on Monday, so I want to rush through it again today. Let's review it. A SimPorter takes at least 2 different molecules, different kinds of molecules, moves them the same direction. Sim means with. So same direction. Hopefully we're going to be SimPorting the next week or so. SimPorting information and SimPorting our biology knowledge. But anyways, so this particular protein, first of all, there is a concentration gradient, which basically means that sodium potassium ATPase, which we talked about on Monday, it's not a SimPorter, but it's an antiporter, is always transporting, what's the ratio of what going where? How many sodiums go in which direction? Sodium, potassium, ATPase, always, always, always transports. Okay, yes, Anna. Boom, using ATP always. Okay, so once it does that, that means that the sodium concentration outside of these cells in general, sodium is always higher outside of cells than it is on the inside. So there's always a concentration gradient for sodium to move into the cell. So this little protein of mine, I'm going to let it shine. This little protein, sodium glucose linked transporter. Okay, try not to get all caught up in the name, but it basically means that it's moving sodium and glucose into the cell at the same time. When sodium moves, there's a concentration gradient, so it just sort of moves. And that energy, you guys have ever been to, revolving door people, where you have to push the revolving door, anyone? Okay? So, you go, and it's a really big one. And you're like, ugh. And sometimes you hit it, and there's like that resistance, right? Oh, I hate this. But once you get it going, it's okay. But isn't it kind of nice too whenever someone else is already moving through, and you're just sort of like, whoop, off I go. And you go in or out. Isn't that nice? You don't have to do the work. Somebody else is doing the work for you. That's kind of what's happening here. Sodium is like that first person who hits the revolving door and starts pushing it. And then Kyle is like, he's smooth as silk. Hey, he just kind of slides right in, and he's like, I'm in this building. Thank you very much. No ATP used by me. So that's kind of like what's happening here. It's like the revolving door. Sodium hits the door and starts the ball moving. Glucose just sort of hops in and goes along for the ride. Okay? Easy. Revolving door. Oh. Bulk transport. Bulk transport. We're moving lots of things in, lots of things out. Two major versions here. Endocytosis, we talked about this real briefly. I mentioned it. It's when, if you've ever seen a video of a white blood cell chasing down a bacteria, it's kind of fun. It hunts it around, finds it. I think I might put that up. And then it surrounds it, and then it pulls the bacteria into itself. And then it eats it. That's endocytosis. Exocytosis is when the cells release a bunch of stuff. Like when, on the quiz, when I said, or we've talked about the hormone insulin quite a few times this semester. Insulin, when those cells in your pancreas need to release insulin, they just release a bunch. Okay? So what does this look like? Endocytosis, two major ways. Phagocytosis. So, cyto, what's that word mean? What's that related to? Cell. Phago, P-H-A-G-O, is from the Greek, which means to eat. So this literally means cell is eating. Yummy, yummy. So there are cells. And actually, this is showing you the plasma membrane of a white blood cell that is bringing in some bacteria. Pinocytosis. Pino is drinking. So that's when you bring in fluids into the cell. Pinocytosis. That's when you bring water or fluids into a cell from the environment. Okay. And by the way, this process right here, so those of you who are in the bio lab, it would probably be most people, maybe everybody. For the Daphnia, there's a question where you have to distinguish whether the Daphnia uses intracellular or extracellular digestion. This is an example of intracellular digestion. The food particle is taken into the cell and then broken down. Extracellular digestion is typically found when you have a gut. The food is broken down in your gut into the smallest component parts and then absorbed into the body. So just to give you a little lab tip. Okay, so Pino means drink. So Phago is eating, Pino is drinking. But when you see the O, or when it's endocytosis, it's coming into the animal. Exocytosis, get rid of it. Get rid of it. Oh, sorry. Still endocytosis. My bad. So receptor mediated. So we have a specific receptors for target molecules. They bind, and then they end up being taken into the cell. That's another way. Receptor mediated endocytosis. I'm not as interested in this kind of endocytosis. And then exocytosis, get the thing out. So oftentimes there's going to be a vesicle. So a vesicle, anytime you see the word vesicle, that means there's a little membrane-bound structure, a ball of membrane that's surrounding some other material. That's a vesicle. In this case, what happens is the vesicle, in the case of if we're releasing a neurotransmitter, then this vesicle would literally be pulled to the plasma membrane. Then what happens is these two membranes, the membrane in the vesicle and the membrane of the cell, they fuse. And then once they fuse, then that vesicle just opens. And the contents are released. And they move away by what process? By simple diffusion. Simple diffusion. So, Marie, did you eat your bagel this morning? No. Who ate this morning? None of my business. I did. And so as that food is being digested, any carbs that are in there, they're broken down. They start entering my bloodstream through my gut. They get absorbed from my small intestine. Sends a signal to the pancreas. We need insulin. Release the insulin. Release the insulin. Insulin transport ready, sir. But this is what happens. The vesicle membrane fuses with the outer membrane of the structure, and it then enters into the bloodstream and diffuses away. It's actually a very simple process. Okay. So, I think I warned you. I sent a message on Schoology that we would be starting Pocket 3 today. Yes? Yes. And I even told you on Monday we'd be starting Packet 3. I even posted Packet 3. So, I guess you don't have Packet 3. I can't help you. So we're gonna move on. And do the best you can. Definitely I know I reminded everybody. And I posted something on Schoology yesterday as a reminder. So we're going to talk about energy. Energy exists in two states. So you can do this old school. You can just write notes down. It's going to be okay. Two states. Kinetic energy. Kinetic energy is when the thing is actually moving. So that's energy in action. Energy in action. Objects in motion can produce work by causing other objects to move. Energy in action. As you're writing, it's kinetic energy. Potential energy. This is stored energy. Potential. It has the potential to do work, but at this moment it isn't. So an example I oftentimes like to use. Let's try it. So right now this is lots of potential energy. The wheels are locked, that increases the potential energy. to be beaten by some stupid whiteboard that doesn't move. Okay. This is high art here. This is a hill. This is a house at the bottom of the hill. This is a rock at the top of the hill. So, rock at top of hill. Potential energy or kinetic energy? Kate. Rock on top of hill. Kinetic energy, potential energy. Just sitting there, it's potential. Good. What if we had rock here? It's still on a ledge. Kinetic or potential, Rebecca? Okay. Which of these two rocks has higher potential energy? The one on top. The one on top. Yes, the one on top. Okay. This is going to be important in a minute. It's only once as they say, once the ball gets rolling, right? Let's get the ball rolling. Once the rock gets rolling down the hill, now it's no longer potential energy, but it is kinetic energy. And its potential is to kaplow the hill, the house down here. I hope you had avalanche insurance. That's the same way. Need some ATP. That's the same thing with these systems. Can hear you. So, energy has everything to do with what's called thermodynamics. These are really fancy words, but thermodynamics just basically means changing energy states. That's all it means. Just means changing energy states. And the first law of thermodynamics, by the way, is this whiteboard in your way over there? Yes, it is. Okay. That would be when you raise your hand. Use your potential energy to become kinetic energy. Dangerous stuff. It's a whole lot of potential energy that nobody wants to see. Okay. It's called Hulk. Anyways, first law basically says that energy can't be created or destroyed. It's only changed from one form to another. We are all energy. Basically, it's what life is. Controlled state of energy, held together. So the whole energy in the universe, ooh, big idea. Doesn't change. It just changes from, it doesn't increase or decrease. It just changes forms. An example, bagel. Your bagel that you eat, that's energy. But it's energy in a form that you can't use right now. But what's the term that we use to quantify the energy content of our food? We call it a calorie. And calorie is actually, that's a unit of heat. That's a unit of energy. So, Marie, when you're cold, you think, oh, I'm going to get out 3 bagels. They all have 200 calories each. I'm going to put them there and I'm going to warm my hands with them. No. Because what do you need to do to those bagels to actually get some heat out of them? What's that? You have to break them down. Soak them in kerosene and light them on fire. You were supposed to do that today, dude. That's why I'm here. Anyways, okay. Second law. Systems are constantly moving from a state of order to disorder. dorm room. Consequently, no process is 100% efficient. Some energy is always lost in the form of heat. And it takes, uh-oh, I just stepped on the thing. Can you still hear me out there? You still hear me out there? Uh-oh.
Yes.
Okay, good. Thank you. I need the feedback. All right. Basically, things are always falling apart. They're always falling apart. It takes a tremendous amount of energy to keep these systems at homeostasis. So all the energy that we use to move molecules across membranes, that's all there to keep you in homeostasis, to keep you in order in some way. This system falls out of order enough, chaos. So we see systems, people in pain, people hurting, people pushed down, right? Out of balance. Pain, chaos. You see the response in society. Same thing happens in us, in our bodies. Okay, there's a correlation here. But anyway, so it takes a lot of energy to hold these systems in order. So our molecules, our bodies they store potential energy in the form of bonds. Because bonds, especially covalent bonds, what are the players? Electrons. Oh, here's a little review. What does hydrolysis do to covalent bonds? Breaks them. Requires the input of what? Water. Releases what? Energy. Remember, this is biology. This is the biology version of all this stuff. Chemistry sees the world slightly differently. Dehydration does what? Forms covalent bonds. What's it require the input of? Energy. What's it release? Water, right? Okay, we've done that one 300 times. That should be automatic. So potential energy, very simply, we're trying to keep this at a very simple level. That's about how I understand things. We're going to store it in the molecules in the form of bonds. And the principal players in forming the bonds are electrons. Yo! Sharing, donating, etc. Electrons. And they're the ones that are moving, right? So they have a lot of kinetic energy, actually. So, we're going to see some reactions. There are reactions that are involved in the movement or sharing of electrons between molecules. Right? This is important. So again, this might be a review for a lot of you, but let's do it anyways. Potential energy is stored in molecules in the form of electrons. And if in the process of a reaction, a molecule donates an electron to another molecule, it's giving it energy. It increases the energy of that molecule. So these are, we've probably heard of oxidation and reduction reactions. Have we heard of those before? Oxidation and reduction reactions? Yes, no? So on the left, the gray little molecules here. So we see a reaction here going from left to right. And the products of this reaction are A plus and B minus. And we notice here that as part of this reaction, A gives an electron to B. So that's why the product then, we see A now has a plus charge and B has a minus charge. That should make a lot of sense for people. So this process, so if molecules are in a reaction, and as a process of this reaction, a molecule loses an electron, we call that oxidation. So when a molecule loses an electron during a chemical reaction, we call that oxidation. B, we said, gained an electron. So if in the process of a reaction, a molecule gains an electron, we call that reduction. Seems kind of weird. Well, if it's reduced, why does it gain Oh, its charge is reduced, maybe. Now, the other thing we need to see is that because A has lost an electron through oxidation, it's now A plus is now at a lower energy state. It gave its energy to B. B minus is in a higher energy state than B. So there's a couple of mnemonics. Oil rig, oxidation is loss of electrons. Rig, reduction is gain of electrons. Or another mnemonic is Leo Szesger. Lose electron oxidation, lower any energy state. Gain electron reduction. Did I say lose electron reduction? Lose electron oxidation. Brain didn't work. That is now lower energy state. Gain electron reduction, higher energy state. And these processes are always going to happen together. Together. So the reactions then, wherein one molecule is oxidized and one is reduced, are called redox reactions. Redox reactions. Okay? Just to sort of rehash. A plus is in a lower energy state than A. It lost an electron. B minus is in a higher energy state than B. It gained an electron. So as long as you remember that electrons have a lot of kinetic energy, and you follow that movement of that electron, that's all you have to follow, and you know which one has lost energy, which one has gained energy. Just follow the electrons. Follow the electrons. Now, all of this is to set up for everybody's favorite processes of glycolysis, pyruvate oxidation, Krebs cycle, and the electron transport chain. What is all that stuff? Well, that's how we extract and transform glucose to generate ATP. But metabolism is the total of all chemical reactions in a cell that are related to sustaining life. And there's two big categories of reactions. And we identify what kind of reaction it is by the end product, for the most part. All right? By the end product. There's anabolism and catabolism. There's anabolism and there's catabolism, as opposed to cannibalism. We don't talk about that in this class. Cannibalism. Hey, but wait, I did coat myself in kerosene. Let me try. No, all right. So, anabolic reactions. Processes that, and you have this text is in the note packet. But basically, if we begin with monomers and they undergo dehydration synthesis reaction to form polymers, that's an anabolic reaction. We're starting with monomers, we end with polymers. Start with monomers, end with polymers. So, glucose to glycogen, or glycogen to glucose. Which of those two is the anabolic reaction? Emily. Glucose to glycogen or glycogen to glucose? Which of those two is the anabolic reaction? We want to go from monomer to polymer. What's that? So are we ending with glucose or ending with glycogen? I'm sorry. Well, I'm sorry. It's the Mac. I can't. Marie? Glucose to glycogen. Right? Glucose to glycogen. Amino acids to proteins. Anabolic reactions. Catabolism. Processes that release energy by breaking covalent bonds in polymers, resulting in the release of smaller units, monomers. So we're going from larger to smaller. That's catabolic. A catastrophe. Everything sort of gets destroyed. Catastrophe. Kata means thrown away from you. Like a catapult. Shoo! So you're breaking things down. So when we break down glycogen and we release glucose monomers, that's catabolic. We had an example of the mitochondria going into the lysosome and being digested. That's a catabolic process. It's breaking it down to its simplest components. And when we talk about breaking down glucose to ultimately form ATP, we're going to cut all those covalent bonds that hold those carbons together. That's also going to be a catabolic process. Yo, hey, that's me. What? Sometimes that's me when I grade quizzes. I'm just kidding. Sort of. All right. What we're going to do is we're going to take the baked potato and we're going to put it in that mouth. Think it'll fit? Yo! So, first step. We're going to eat the baked potato. What do you think? Anabolic? Catabolic? Anabolic? Catabolic? Don't careabolic? Hyperbolic? Parabolic? It's okay, sorry, it's catabolic, all right. Oh, which of these two, this is a great review question, Father Sean, thank you. Which of these two would we likely find in our potato? Glucose or amylose or amylopectin? Christy? I don't know, that's a good answer. One of these two is found in plants, one of these is found in animals. Jared? Amylose or glycogen? Glycogen. You've got a lot of glycogen in you. Okay, amylose, amylopectin. Another name for it? Starch. Okay. So we break this down. After you eat, you begin digesting it. You break it down into these little glucose monomers. So this process, anabolism, catabolism. Anabolism, catabolism. Don't careism. You shouldism. Okay, it's catabolism, breaking it down. You go out for a walk. You burn off some of these, like 1, 2, 3. All right, you burn off 3 of those to make ATP. We're going to talk about how that happens. But then everything that you don't burn off, 1, 2, 3, these 4, they get now combined, and we begin to build our glycogen molecule. So what kind of process is this? Anabolism? It is our catabolism. No, because we're going from smaller to larger. Good. Okay. That's it for today. Bring your packet on Friday, packet number 3. We are cruising. We are at cruising altitude. We are going to complete. We're going to do a whirlwind. And by whirlwind, I mean like 5 minutes. Highlights of Gibbs free energy. 5 minutes. That's it. That's about. And then finish. We're going to finish all of page 8. And I would answer on page 9 up through pyruvate oxidation on your study guide. Finish all the questions for pyruvate oxidation. We might even get to the Krebs cycle on Friday. We are grooving. See you next Friday. See you Friday. If I could once again have my cleanup volunteers, I'd like it. Thank you very much. Thank you, thank you. No, I sent an email last night.
Okay.
Did you get the email?
Yeah.
Everybody starts at 2.
Okay.
Everybody starts at 2 today.
But we're still online.
Yeah. We're not coming to class. Yep. We're just online. Everybody together. All 22 of you.
Okay.
And wherever you want to do that, if it's in the library, in your room, wherever, it doesn't matter.
Okay. Sounds good.
Yep. See you, Marie. So John, do you still want to? Oh, wait. Let me turn this off. You do your thing.